Good morning, everybody. Let's wait a minute or so for people to roll in. I remind myself where we left off. Okay. Pardon me. Doobie doobie doo. Anybody got any questions in the meantime? I'm all ears. Oh, actually, let me just go close my office door. All right. So we have linkage today. Fun stuff. Okay, so get your maths heads on, because we'll be doing probably before the end of the class today, but certainly on Friday, we'll be doing a ton of math. All right, so get my thoughts together. Basically, what we're talking about here is, just to recap, the process during, prophase 1, right, meiosis, when homologous chromosomes line up and they swap bits of chromosomes to kind of hold them together, for one thing, but also to shuffle the alleles that you have, right? So essentially what you get as a result is 4 different chromosomes. So remember, one cell entering meiosis ends up as 4 haploid cells, haploid gametes, right? In the case of sperm, or, kind of 4 potential gametes in the case of oocytes. And each one of those gametes will have a different chromosome in them, right? So these are all homologous chromosomes, pretty much, right? Well, they are homologous chromosomes. So imagine that each one of the daughter cells has one of these, right? And you can see that if the original parental homologous pair is heterozygous at 2 spots, right? A and B. So the blue one is recessive, the red one is dominant. By the time you go through the whole process of meiosis and recombination and reductional division and so on and so forth, you end up with 4 gametes that have a different combination of alleles in each one of those cells, right? So you have 2 gametes that have parental or non-recombinant chromosomes, right? The blue one here is the same as the one up here. The red one down here is the same as the one here. And 2 where you have a different combination of alleles, right? If you remember when I was talking about this a way back ago, this is like if random alignment on the metaphase plate is shuffling a deck of cards, right? Essentially, this is like tearing a little bit of a card off and sticking onto its matching card on a different deck, right? And so this is a way of like fine scale rearranging and shuffling the allele combinations, right? We're not generating new information here, right? We're simply taking the information that is present in that individual and just finally shuffling it up so we get different combinations of those alleles already present in the offspring. So that's kind of like the purpose of recombination, or one of the 2, right? The other one holding those homologous pairs together. The other is a tool, right? So a tool for us, right? And we can actually use this to figure out how far apart genes are or loci are on chromosomes, right? So again, can't emphasize this enough. When you look at two different traits and the genes that are responsible for those traits are linked, right? So essentially they are close enough that occasionally no crossing over takes place, right? Again, so if you want to write something down, write this down. Linkage means that recombination does not always occur between two genes because they're close enough that just by chance, sometimes it doesn't occur between them and occurs outside of them, right?
Can you repeat that one more time?
Sure. So linkage is when this crossing over recombination doesn't always occur between two genes. And so if you flip that round, I can even write this down on the chat box. Doop, doop, doop. And if you flip that around, essentially what that's saying is that two genes are close enough, right, physically on a chromosome that the probability and again, recombination is random. Not RNA, sorry. Got my A's and N's the wrong way around. Right, so essentially the basis for this, like the actual mechanistic basis, is that recombination is random, right? And so for every given like unit of chromosome length, you have a certain probability that recombination will occur there, right? And so this is almost like the kind of additive probability, right? The exclusive events deal, right? If you have twice the distance between two genes, you have twice the probability that recombination will occur. If you have 4 times that distance, you have 4 times, right? And so on until you get to those genes. Shoot, you can't even see my hands. There's one, the other one's over here. You get those two genes far enough apart that recombination will always occur. When that is the case, those genes are not linked, right? So if you have a case of B, right, where you always, every round of meiosis without fail, you always get an equal number of recombinant and non-recombinant chromosomes, those genes are not linked. It's only when you get an excess of these non-recombinants, which occurs simply because crossing over doesn't occur, right? So if crossing over doesn't occur, you don't see those recombinants. They don't appear, right? All you get are parental non-recombinants. That will only occur when, just by random chance, crossing over does not happen between these two genes. It's not that it doesn't happen at all. That's a really important point to make. It's simply that it doesn't happen between those two genes. So for example, unplug my laptop. Oops, clang. Where's my pen? Ship it. Actually, sorry, I've been told I shouldn't swear when I'm talking to students. Got to be professional. All right, let's get my. Actually, you know what? Someone once said that it works really well like this. So let's do it this way instead. Make sure you can see what I'm drawing. Doesn't actually matter whether you put them up and down or side to side, it's exactly the same thing. Let's say plus, plus, and A and B. I'm not writing them for both of them just because it kind of gets messy. Right. So essentially you're going to get a crossover event. You can get a crossover event here, right? That will give you four chromosomes that look like that. Can you see what I'm writing? Not sure if the glare's an issue or not. So recombination has occurred, but it hasn't occurred between those two things, right? For that to occur, you'd have to have instead something like this. Right? You'd have to have recombination occur here. Here. If I was South African. Here. Right? And so that would get you essentially, let's make sure I draw it right. That, which would be AB, B, A's up there, plus, 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 plus. Right? Only if you have recombination occurring here. Does that make sense so far? What I'm trying to get across really is that this is a random process, and any random process, right? Like, for example, walking under a tree and getting a bird poop on your head, which happened to me the other day when I was walking my dog. Totally random, right? X probability of it happening. Obviously, the more time you walk under trees with birds sitting in them, the more likely you are to get poo on your head. Right, so this, hang on, let's take a picture of this. Oh, I need to show you something cool. My daughter's crushed it in whiz. Isn't that adorable? It's my Christmas present from a couple of years ago. It's a giraffe. And I'm not sure what that is. I think that was when the epidemic started. Anyway. Yes, my daughter, you can actually see her. She's, fingers pointing, that one. No. That one. There you go. Yeah, it might be me. Hair's a little bit too long there. Anyway, all right, let's take a picture of this just before I rub it off. Okay, now the other way of doing this, right? So if those genes are that distance apart, there's the probability, based on how far apart they are, that this is what happens, right? You get a recombination, I've just rubbed it out, which is annoying. You get a recombination outside of the two things, right? So now what about if we have these two genes really far apart, right? Let's say they're as far apart as you can get them on that chromosome arm. Right, so one is like bang up against the telomere up here, the other one's bang up against, ouch, the centromere down here. What do you reckon the probability is that there'll be a recombination event between those two? If you always get a recombination event on the arm of a chromosome, what do you reckon the probability would be? It will definitely take place every single time, 100% probability. So you'll always get a recombination event between them, and you'll always get an equal mix of parentals, which is this one. Hang on, we can do that with a P. parental. That's terrible, Ryan. And recombinant chromosomes, always. So you can actually even see, this is recombinant. You can take that to the other extreme, right? I mean, the reason why I want to get this through is because this is a really kind of foundational principle, oops, behind this, behind what's going on. Yeah. What about if we have go a little closer, right? Those two genes are so close together, let's say they even overlap a little bit, right? What's the probability now that you'll get recombination happening between those two? If they're effectively like right next to each other. Not just not very high, Sophia. We need to be more precise than that. If they're so close together, you cannot fit a cigarette paper between them or some other very, very small thing. It's not going to happen. No recombination is going to occur. So you'll always get the parental non-recombinant chromosomes out of that, right? Yeah, make sure I'm pointing in the right direction. And so this, basically, I forgot to write out on the other one. These are completely linked, absolutely and totally. These ones are unlinked. No linkage is occurring. Essentially, what's happening is these two, I'm Zacking. Thankfully, I've got a fairly light computer, right? These two genes are so far apart that effectively they're behaving as if they're on different chromosomes. Remember the whole 7p traits that Mendel looked at, which by chance happened to be on different chromosomes, right? Effectively, these two genes, even though they are on the same chromosome, they're behaving as if they're not, because you'll always get, actually, let's put it up here, make it even clearer, a 1:1:1:1 ratio of those chromosomes, always, which is exactly the same as if those genes are on different chromosomes, right? If you look at the PowerPoint kind of further up, it shows that, right? Now, what you'll get here, actually, let's move that down a little bit. I should put 100% linked, right? Now you have essentially a 1:1 ratio of parental to parental. If you were to write it the same way up here, if it was P, R, and R, right? Down there, run out of space, you would be 1:0:0:1. Because these recombinants are absent. You'll never see them. And I've actually done crosses with, you actually can use that. Not perhaps quite this extreme, but a lot of the worm crosses that I do, you can get markers, linked markers. So someone does the hard work of getting a gene which has a visible phenotype when it doesn't work, say a short and fat worm or a worm that can't go backwards or something like that. And they cross it and they get recombination, very, very recombination. So that gene is now linked to the thing that you are interested in, right, in your particular gene. And so when you do a cross, because they're so close together, you can make a pretty decent assumption that they're not going to get separated, right? So you look for one and you'll get the other one, right? And that's a lot of what I do, is doing crosses with those linked markers, because it's a lot easier looking for those than it is the phenotype of the thing that I'm interested in. All right, how are we doing with linkage? Does everybody get the idea of unlinked, far apart, 100% probability? Linked completely, 100%, so close, 0 probability of recombination, right? Does everybody get that? Yum. Awesome. Now, hey, excellent. Clear as mud, as my dad would say. Now, the thing is, those are the two extremes, right? So far apart that they're unlinked, so close that they're 100% linked. What you'll actually find, however, is most are somewhere in between, right? And so if you have genes which are essentially partially linked, right, you'll see a reduction in the amount of, pardon me, in the amount of recombinants. They're not going to disappear entirely, but you'll just see very, very few of them. All right, so essentially what you'll be seeing in your fly lab. So this is a, if I can get to it, good example, right? So this F2 generation should be 9 to 3 to 3 to 1, right? That's what you would expect to see if these genes were not linked, if they were assorting independently, right? However, as you can see, these two non-parental recombinations, right, that are not the same as either parent, they are present at much lower rates than you would predict if they were not linked, right? So this is telling you that there's linkage going on. It's not complete, right? Because you do see these two non-parental combinations, but there is a vast excess of the parents, right? Essentially, the phenotype combinations you see in the parents, you see way more of those than you would expect to see if linkage was not occurring, right? So that is the classic symptom of linkage. Let's see, get the chat box up if I can. So always kind of always go back to the Mendelian inheritance. Again, that's why it's a useful kind of thought construct to work with, right? Because that's telling us what we should get, and obviously we don't always get that, and so that tells us that something else is going on, right? In this case, if you still get 4 different combinations, but there are far fewer of the non-parental and far more of the parental combinations, that's telling you linkage is occurring, right? Quick question, recap. What would you say is occurring if you didn't see one of these phenotype combinations at all, but you saw 3 of them? What would you tell me is happening in that situation? Or maybe you're seeing only 2 phenotype combinations in the parents and only 2 phenotype combinations in the F2 generation, even though you have 2 genes. No, not linkage. Something else entirely. We covered it in the non-Mendelian inheritance lectures. Begins with an E. When one phenotype at one locus is masking the phenotype at another locus. Ends with S. Epistasis. All right, good. So this is not epistasis, right? Because we are seeing all 4 different phenotype combinations, as you would expect, given that there are 4 different phenotypes in the parents, right? This is linkage because even though we see the 4 different combinations, they're not in the ratio that we would expect based on Mendelian inheritance. Okay, so just kind of keep that in mind. One way of thinking about these, which may help, depending on where your mind works, may not, is thinking about what are the key characteristics, right? What's the key characteristic of incomplete dominance? Key characteristics of codominance, key characteristics of epistasis, key characteristics of linkage, right? So that when you look at a question in the exam and you see blah, 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 right? You can go, ooh, I recognize that and that he must be talking about such and such. Okay, I'm working with this kind of problem now, right? And then that makes it a lot easier rather than having to figure everything out from scratch. Okay, so a little bit of terminology. So you can only figure out what, if we are talking about linkage, you can only figure out what kind of configuration those alleles are in by looking at the offspring. So don't assume that we always have both dominant alleles, both recessive alleles on the same chromosome, right? So dominant, dominant, recessive, recessive, right? So that the recombinants are a mix of those two, a dominant recessive, a recessive and dominant. Do not assume that, right? Because that may not be the case, because you can have that in a different way, right? This is called trans configuration. This is cis configuration, so cis meaning the same, so you have the same kind of type of allele on the same chromosome, so dominant and a dominant. Trans being opposite, we have the opposite type, right? We have a dominant and a recessive and dominant. So now, when you look at the offspring, now the recombinants will be dominant, dominant, recessive, recessive, right? So don't be tricked by that, right? We're not trying to trick you, but you have to not assume anything in regards to configuration. Gene order, right? When we get 3-point crosses is another thing that you can't assume, right? You actually work that out. So remember, cis versus trans, you're not going to tell unless you look at which combinations are lower in frequency, because those are the combinants, those are the configurations that you don't see in the parents. Okay, so again, this is, I'm not sure if you covered this in Mr. Penny's lab, but this is how I started writing stuff in the fly lab, right? Actually, this is kind of the opposite way around from above, which is a little bit annoying. But anyway, we write the genotypes with plus being the normal and a letter for the mutant or the recessive allele, and a line basically showing, right, you know, we're talking about the stuff above that line is one chromosome, stuff below it is a different chromosome, right? So if we have trans, right, we have W plus plus M. If we have cis, we have both the same, WM on one and plus plus on the other one. Okay, those are just kind of terminology and notation type deals, language stuff, essentially. Okay, so What we're going to get into now is working out numbers, right? Actually using this as a tool, right? And so now what we're going to do is work out the recombination frequency. Okay. So, oh, I better take a picture of that. Scrub this off. All right, so here again, we're going to talk about, let's just get rid of this so I can bring that up elsewhere. Okay, so make sure I can actually, there you go, that'll do. So I can make sure I can point the camera at the right thing. Okay, so we have 4 different, this is going to be between 2 genes, right? So this is 2 different traits that we're looking at, 4 different phenotypes. We have, normally, okay. Aha, I get to practice my fly drawing. Ba-dum-ba-dum. Aha. Sure I can see it. Yeah, isn't that beautiful? All right, so we have eye color and wing size as our 2 traits. We have, for the wing size, we have normal size wings, those big ones, and these cute little miniature wings, which presumably are not very good for flying with. For eye color, we have the same as what we're working on in lab, white eyes and red eyes, right? Red eyes are plus, and miniature wings would be, I don't know, W, right? No, that's going to be M actually. Make sure I get that right. Okay, so just I'm looking at the numbers. And all right, put some numbers up. So, which of these are the parentals and which are the recombinant? Well, actually, hang on. Before I even get further, is linkage occurring? Yes. Why?
Because there's more of the parentals occurring.
Right. Or another way, which is like the kind of the flip side of that, is that you don't have equal numbers roughly of the offspring. Right. And so which of these 4 are the 2 parents?
The 2 on the left.
Right. I'm actually quite proud of my flies. I'm getting good at this. Not quite as neat as cats, but good enough. Right? Which means that these are recombinants. Yeah, does that make sense for everybody? So total is, actually, I'm going to cheat and look at the PowerPoint. $644. Now the recombination frequency is how often you get recombinants out of the total, right? So that's going to be these two added together, which is 216 over 644, which again, I'm just going to cheat. Oh yeah, go away. Dum, dum, dum. 33.5%. And so we can just give that a unit, which is not arbitrary, but it's kind of a theoretical unit, which is centimorgan, after Thomas Hunt Morgan. That's our mapping distance. Does everybody understand how I got from this to this? Cool. You always take the number of recombinants and divide that by the total. Actually, you need to do times 100 as well to get percent. You get the idea. Now, in terms of genotype, right? Red eye is wild type. So this, well, the gamete that that it got was essentially that wild type, and the miniature wings. Is over I think it's over Y. Similar kind of cross to what we're doing in lab. This one is white-eyed plus, right? So the recombinants are now. What configuration are these two genes in terms of linkage in the parents? Cis or trans? Anybody remember? Kind of cast your mind back up. Yeah, they're in trans, right? Because you have, these have got to be the parents, and you have one of each. You have a mutant and a wild type. In the other one, you have the opposite, wild type and mutant. And only in the recombinant offsprings do you have the same dominant or recessive, right? So those are in trans. Awesome. Okay. Oh, and actually, just before I do that, this, go away. Right. And so what this is telling you is that essentially you're going to have, what's that divide up into? 16.75% of the gametes are going to be this. 16.75% of the gametes are going to have this allele configuration, and the rest is, oh, I have to do math in my head. What's that? 32.25. So the remainder, which is 66.5, oops, not very good at doing math. There you go. The remainder of the gametes are going to be split between these two, right? So we have about a third each of all gametes are going to be either that combination or that combination, right? So you can work back from those numbers to actually figure out what's happening in meiosis. What are we actually seeing at the end of a full round of meiosis? Here we go again. And that's essentially what this question is about, right? So if we have a recombination frequency of 16%, right? So that tells us we have 16% of our offspring, are recombinant. What percent are parental for a start? If we have 16% recombinant, what percentage parental? No. So recombinant are our two different combinations. Total, that's 16%. The other two total combinations, the parental ones, are going to be 100% minus 16%, right? Which is 84, right? So if we want to figure out in turn what the percentage of each gamete is, right? Now you get it. Now you would be correct for the recombinant. The two different recombinant gametes are going to be 8% each, because you add those together, you get 16%. So remember, every round of recombination will produce two different combinations, right? They're going to be mirrors of each other, right? If you cross over, you're going to get essentially the end of one at the end of the other and the reverse, right? Doesn't matter whether they're in cis or trans. All that tells us is how the alleles are present on the different chromosomes, right? So it doesn't, in terms of actually the recombination frequency and percentage recombinance, it makes no difference whether they're in cis or trans. However, it does make a difference in terms of what the recombinants will look like, right? Versus what the parents will look like. You can't assume that. You have to work that out from the recombinants or from the parentals that are in excess. So the cis versus trans thing is really figuring out which chromosomes have which alleles. The recombination frequency is how far apart are those two genes on that chromosome. And to figure that out, the cis versus trans doesn't actually matter, kind of. It really only matters in terms of figuring out what the recombinants are. And you do that by finding the ones that have the lowest percentage. Does that help? Yeah. Don't get too tied up on the cis versus trans. That's really more figuring out gene order and allele combinations, right? The thing in many ways, what recombination frequency is another piece of information, essentially, right? The two don't have to go together. Okay, so now we have another one, right? So now we have diminutive males, or just like teeny tiny males, and white-eyed females, right? So again, what's the configuration here? Is this in cis or trans? Trans. So if you know that, what combination of phenotypes would you expect in the recombinants? So if we have mutant, wild type, mutant in the parents, what combinations would you expect to find in the recombinants, the non-parentals? Cis, right? So you would expect to find wild type, mutant, mutant together, right? So that can actually help, you know, if you're given that information ahead of time, right? If you're not given that information, you look at which of these are the recombinants, top two or bottom two? Which flies are the result of recombination? Top two, bottom two out of these four?
Bottom two?
Right, exactly. Because they're present at far, far lower frequency. Now compare this to the other one that I just put up on the board, right? Do you think that white and DM, the genes, are closer or further apart than white and miniature wings, right? Just to refresh your memory. Make sure you can see, right? Which genes are closer together? Remember about probability of recombination is random. Which genes are closer together? These ones that we're looking at now. Sorry again?
They're closer together.
Which ones?
The white eyes and normal size, and then the red eyes and diminutive size.
Yeah, oh, so those are phenotypes, remember. So the genes we're talking about are the white and the DM.
White and the DM.
Right, exactly. Awesome, right? They're far closer together, 10 times closer together, actually, if you want to be somewhat mathematical about it, because you see far fewer recombinants. So you can either do that by working out recombination frequency, right? So the map distance of the ones we did on the board was 33.5 centimorgans, which is a measure of distance. These are 3.1 centimorgans, right? So these two genes are much, much closer together than the white and diminutive wings, or white and miniature wings rather, and white and diminutive males. Right? So that gives us, you know, well, so the reason why we know that's the case is because the probability of recombination occurring here is lower than it occurring here, 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 or here, right? If those two genes are further apart. And so now we can actually start figuring out, well, where are these genes, like relative to each other on a chromosome? And so we can figure that out for each pair of genes, right? You know, and we have another example here, right? And this is something that I want you to do, because we only have a few minutes left, between now and Friday, right? And we'll cover this at the beginning of class next Friday, but I want you to try it on your own first, right? Actually, there's the answer there, but that's, I'm not going to tell you that. Don't look ahead, right? But if you have 3 different genes, right? Actually, I should work out some data for this so we can do this as an example with those examples, right? If you have 3 different genes, oh yeah, that's something else. You can do that individually, right? You can do a 2-point cross between, say, white and diminutive males, and you can do another 2-point cross between white and miniature wings, right? The question is white in the middle, like so? Is white in between? Where is white, right, relative to those different genes, right? We don't actually know by doing a 2-point cross. And so the best way of doing that would be to do a 3-point cross, right? Where we do a cross with, we're looking at 3 different genes at the same time. So instead of doing white versus diminutive males and white versus, I'm trying to kind of use scale, white versus miniature wings, now we're going to do a cross with white, diminutive males, and miniature wings as things that we can track, right? And so the plus side to that is A, it takes a lot less time, right? And also you don't miss these double crossovers, right? So you can potentially get 2 crossovers between your A and your B. And if that does happen, you miss those with just a 2-point cross, right? And so 3-point crosses resolve gene order, they're more efficient and they're more accurate. And so what we'll do at the beginning of next class is look at this 2-point cross example, right, which uses actually 4 different genes or letters. And once we've done that, we'll start on doing 3-point crosses, right, of which we have multiple examples here, and I'll give you some examples. Actually, I'm going to just make a note to myself to look up diminutive males, white, and miniature wings, so I can actually get some somewhat real life map distances, work up some numbers for you. All right, so there's a lot of math going on here. It's once you get the idea of what's going on, it's actually fairly straightforward. It's kind of like doing a chi-square test or a dihybrid cross or any of that stuff, right? The laws are pretty straightforward. You just have to understand what you need to do and what order you need to do it in. All right, so next class, we'll spend a little bit of time on that 2-point cross example, and then we'll go into 3-point crosses and we'll do a ton of work on that. So do your homework, read again if you haven't already, but even if you have, do it again anyway, 3-point crosses, and see if some of that twigs. And whatever doesn't, we'll cover that on Friday. Okay? Cool beans, everybody. I shall see you either in an hour or so, or I'll see you on Friday.